right, some great poster pitches. On to our second speaker for this morning, Dr. Julia Domingo. Dr. Domingo completed her PhD work with Dr. Ben Lehner at the Center for Genomic Regulation of the Barcelona Institute of Science and Technology in Spain. Currently, Dr. Domingo is an EMBO postdoctoral research fellow in the lab of Dr. Tuli Lapalainen at the New York Genome Center, studying how to integrate deep mutational scanning and single-cell technologies to better understand how the non-coding genome regulates gene expression. Today, she'll be talking about her work, Global Mapping of the Energetic and Allosteric Landscapes of Protein Binding Domains. Dr. Domingo.
Thank you for the kind introduction. So yeah, my name is Julia. I'm a postdoc in the lab of Tuli Lapalainen, but today the work that I'm going to talk about is mainly involved my last years at the lab of Ben Lehner where I did my PhD. And this not only involved me, involved many people. This project was led by Andre and Jörn and me, and we had help from Guillaume and Christina too. So I'll start by posing the big problem that motivates this project. And this is that even has passed many, many years since we decoded the genetic code, and now this is a very trivial thing to do, to go from DNA to any amino acid sequence. This is a very simple task. Going one step further is much more difficult for us. So for instance, saying how amino acid sequence encodes for biophysical properties of proteins, it's a complicated thing to do nowadays. And more if we want to know like how a particular amino acid substitution can affect binding affinity, the stability of the protein, if the protein will aggregate, etc., etc. And this has severe consequences. The first one is that we are not very good at being able to predict what's going to happen. And nowadays, most of our favorite disease genes have a lot of variants that are of uncertain significance. The second consequence is that we are really bad at engineering biology. We cannot do it in the same way that we engineer cars or bridges, and usually takes us a lot of time, a lot of money, and involves a lot of brute force. So we wanted to dig a little bit more into this step, like going from amino acid sequence to biophysics, so doing this from sequence to energy step. But this, again, there's many years that we know how to decode the DNA, and why is this not a trivial thing to do? There's many reasons for that, but I want to highlight one of the reasons with this toy model. In particular, we were interested in binding domains. So in a protein that can bind another protein, we can think of this of a three-state equilibrium model where we can have three different states: a state of the protein that is unfolded, and then a second state where this protein is folded but not bound to the protein, to the binding partner, and a third state where this is bound. And as you can see, going from one state to another involves like changes in the energies of binding and folding. Thus, mutations that affect these two parameters can affect the final fraction of proteins that are in the final state of bound to complex. And I want to highlight this here in this heat map where we can, the color of this heat map represents the same, like each unique color is the fraction of folded and bound complex, right? An isochore, delighted here in red, would be one particular state where there's the same amount of folded and bound protein. So we can imagine that from a wild type, we acquire a mutation that brings us to a particular decrease in this amount of bound complex, and this mutation can affect the binding energy, but we can obtain the same exact consequence by just a mutation that affects only folding. And we can even have a third scenario where a mutation affects both of these parameters, right? So this would be a pleiotropic effect. And this is what we call biophysical ambiguity, where we can get the same molecular phenotype, or say the same amount of complex bound, by the combinations of many of these possible effects. So how do we disentangle these ambiguities? One way to do it, and this has been shown before by previous work, is to make use of higher order mutants, like, for instance, double mutants, because if we assume that at the level of energies, these single mutant effects act additively, then this constrains the possibilities of the effects that we combine of folding energy and binding energy. And we cannot do for one double mutant. If we have many, many double mutants, the same single effect will be measured many, many times. That becomes a finite and kind of trivial solution, right? But for that, we need many double mutants, and this has only been shown very precisely for the most complete double mutant data set that we have so far in a binding domain. So these libraries are very complex and troubleshooting at the time of doing experiments, right? So we wanted to find another solution that can reduce this complexity. And the other solution is this one, is to not only measure one phenotype, in this case, the amount of bound protein, but measure something else that will constrain our data. And this is if we have an assay that can estimate or be proportional to the amount of abundance of the protein or the folding, so that we fix that parameter, that constrains even more the data. So that's exactly what we did, and this, it's why we call it multidimensional mutagenesis, because the key of this experiment is going to be assaying the effects of mutations in many different genetic backgrounds and in multiple phenotypes. And we do this, of course, with our favorite experiments at the mutational scanning, where the selection assay, here, it's not one selection assay, but it's going to be two. And we call this double DPCA, because it's based on protein complementation assays. So the first one, binding PCA, in this first assay, we have two proteins, A of interest and its partner B, and we fuse these two proteins to two halves of an enzyme, DHFR, which is resistant to methotrexate. Methotrexate, when we put it on the media, will inhibit the endogenous DHFR of yeast, and this is necessary. The DHFR is necessary for growth of yeast because it's required for synthesizing and replicating the DNA. Thus, when A and B come together in space, they bring these two halves together, and this reconstitutes this methotrexate-resistant version of the DHFR that will allow yeast to grow. So here, whenever we have a mutation that affects binding of A for B or affects the stability of A, this will have an impaired growth in yeast. In the second assay, it's very similar. It's based on the same principle, but in the abundance PCA assay, what we do is we overexpress the second half of the HFR. We no longer have the partner B, so the encounters of these two halves happen randomly in the cell. So now only mutations that affect the stability of A are going to have a deleterious phenotype in the assay, and no longer the ones that only affect binding affinity. So here, to which proteins did we apply these methods? These were the SH3 domains of GRB2 and the third PZ domain of PSD95. And we selected these two domains because these are some of the most abundant domains in the human proteome. And also the second domain was known for allosteric regulation, so we wanted to see if we could capture that. As I mentioned, we create these mutant libraries, including single and double mutants. So we did this in several different ways. And so we have a bunch of double mutants for each single mutant, ranging from a median of 5 to 74. So these are the initial results that we have after we run these experiments. And here I'm just showing it for one of the domains. But as you can see in these heat maps of single mutant effects, there's a lot of red, meaning that most of the mutations are deleterious for these two particular assays. And as you can see in the distribution of fitness effects, we also measure double mutants, which tend to be even more deleterious. But again, these are molecular phenotypes, and what we are interested is in the underlying biophysical traits, the energies. So this was mainly the work of Andre and Jörn. And here, coming back to that model that I explained before, there's a way that we can go from these molecular phenotypes to the underlying physical trait. And we used, in this case, the Boltzmann's probability distribution, which basically tell us, for instance, how to relate a molecular phenotype. In this case, would be the probability of molecules to be in certain state K, for instance, in the state of folded, to the, that relates this to the underlying biophysical trait in a nonlinear fashion, right? And you have to believe me or believe them that this can be translated in a series of equations that will determine the fraction of these molecules in these different states that we will try to fit our data on. And to fit this data, we used neural networks, and this was mainly the work of Andre, that he's developing it developed this method we call Mochi. It's in preparation, and it's going to come soon, so stay tuned for that. But basically what it does is, as an input, it takes the sequences of all these mutants, single and doubles and wild type, and will try to predict exactly the binding fitness effects and the abundant fitness effects. This model has a couple of assumptions. The first one is that the energies, at the level of energies, this combine additivity in double mutants, and then from energy to molecular phenotype, that would be the fraction of molecules that are in these two different states, and there's a nonlinear relationship that is shown here. The only fitted parameters in this model are the linear transformation that goes from this frequency of molecules in these states to the fitness effects, which you know are linearly related, and the actual weight coefficients, which are the energies, the free energies. So how does this model do? And the answer is that it does very well. So we get the correlation between the observed fitness and what we predict. It's extremely high, and it reaches the level of the variance explained that we see in its replicates of the experiments. And just wanted to show you that it's important that we have this multidimensional data, because if we only use the binding PCA data, in this case, just one of the assays, or we just use the single mutants, the accuracy of these predictions are not as good. We also, for the few information that we could gather, we could validate our inferred free energies with in vitro experiments, and they correlate quite well too. So going into the results, here I'm showing on the top the molecular phenotype, and on the bottom the biophysical trait. And there's a few differences, and some of the things that we want to highlight is the first one, that the below map is much more complete, so we have less of these white squares, and it's because even if we don't measure the effect of a single mutant in an assay because it's a low confidence or the library was biased, we can infer the effect of the free energy based on the multi-mutants, right? So it's really good. The second one is that for this case, the effects on binding are smaller and subtle, right, compared to the top one. And this is maybe because some of the effects that we see on the top molecular phenotype are driven by stability rather than binding affinity. And finally, these are in interpretable and meaningful units. So now these units are in kilo-K per mole, which it's much more interpretable than relative fitness. Okay, and this is just to show it in the case of folding, and the take-home message here is that a lot of mutations affect stability, and this is the main driver of disrupting protein binding complexes. But we could ask many other things with this data set. The first thing that we wanted to see is like how many times a mutation can affect more than one biophysical trait, and the answer is lots of them. Although we see stuff that makes sense, that for instance, we see like the amino acids that reside, like, or the residues that are in the binding interface affect strongly binding affinity rather than folding energies. And more than 2/3 of the mutations that we see in these data sets have effects on both biophysical traits. So this can have like consequences at the level of genetic prediction. It's when two mutations combine, then it becomes a big mess to be able to predict that if we don't know the underlying biophysical traits. Looking at stability, we see that, of course, and this makes sense, that mutations that destabilize the proteins are enriched in the core of the proteins, and here you can see this core highlighted in red. But what we notice is that surfaces had this an enrichment for some residues that harbor like mutations that would stabilize the protein, so that decrease binding of folding energy. And we see this in both domains for surfaces and interface residues. When we look at the binding energies, we see that those mutations, again, or mutations that affect binding are located mostly on the interface. And this was also obvious, but it was very nice that we could actually identify this binding interface with any prior structural information. So this could be a very useful way of actually finding where two proteins bind together. But what we were more interested in, it was this, in allosteric, which is regulation at a distance in proteins. So basically, meaning that if one site of a protein has a mutation, a transcriptional modification, a ligand binds to it, somehow there's a switch in this protein that in another distal site of that protein, that's going to change the binding affinity, the function, the enzymatic activity or something. And this allosteric, the name was like termed at the same time that the genetic code was cracked, and it's been long known, and now we know that it's central to most biological regulation, but still it's poorly understood when it doesn't involve like huge structural rearrangement on proteins, which is the majority of the cases. Not even we don't know how it works. We don't know how extensive it is. We don't know how many sites, allosteric sites, there are in proteins, because we have no methods to map them nowadays in a very systematic manner. And we would benefit of that, because now we know that the best drugs or the most effective drugs in the market are those that target allosteric sites. And this is because allosteric sites evolve very different than they do binding interfaces or active sites. So they are very specific to each protein, and they are not shared from proteins of the same family. So they are highly specific, and they have very little side effects. So how do we map allosteric sites? So we just look for those residues that have an average that their mutations affect binding affinity to the same extent that they do, or the mean of the residues in the binding interface. And following this strategy, we map two residues in the GRB2-SH3 domain, but these are residues that are very close to not directly contacting the ligand, but on the second shelf of residues after the binding interface. But on the case of PDZ, which we knew that there was some sort of, it was described for allosteric and regulation, there's many more residues, and some of them even form this chain of residues that go from the binding interface to the opposite side of the protein. And we validated that the previous sites that was described in this protein are enriched in the ones that we find, and we find even more sites. So that this is exciting. We also look not for residues that are major allosteric sites, but if any mutations that we see in these proteins have the potential to tune binding affinity. And when we do this exercise, we realize that many mutations can tune binding affinity, increase binding affinity, or decrease binding affinity. And this involves roughly 50% of all the exposed residues in the proteins. So this is encouraging if we think that now we can, design new drugs targets and to target these surfaces. We had a little bit of an intuition of how the mechanism that may be driving this, and we noticed that the number of allosteric sites decreases when we go far away from the binding interface. And this may be because this is mediated by perturbation propagations effects. We also, something that makes sense with this hypothesis is that glycine residues are enriched for allosteric mutations, and allosteric mutations tend to be enriched in prolines. So with this, I'm reaching at the end of my presentation. I hope that I convince you that doing this multidimensional mutagenesis is a way to, disentangle these biophysical ambiguities. We can use neural networks to go from this sequence to energy space, and hopefully this will bring us to be better engineers, be better predictors of clinicians, and maybe one day to have this complete allosterone map. With this, I'm going to say that if you want to read more about it, this was published a couple of months ago, so check the manuscript. Also, all the reagents and plasmids are available, and of course the code. And with this, I'm going to thank again the people involved and the entire Learner Lab, which is I know that they are hiring. So if you want to know more, like text me or come find me. It's a great lab. I highly recommend it to do a PhD or a postdoc there. And if you're still not convinced or want to know more cool stuff about the lab or what they are doing, check the posters of Albert, Chenchun, and Taraneh on the 16th. And with that, I'll thank you for listening. I'll be happy to take any questions.
Hi, Colin Kretz from McMaster University. Very nice talk. I was curious about the assays that you're developing. There are two assays that you're combining into one sort of parameter. And I was thinking that these two assays probably have different dynamic ranges in terms of the sensitivity pickup, right?
Mm-hmm.
So how do you combine those two parameters to create one value without one assay sort of taking over the determination of delta G?
Yeah, they have different range, and this will depend a lot also on, for instance, the binding partner, if that binds very strongly to your protein A, or if you need like a higher expression or a lower expression. This is something that can be tuned using different constructs, for instance, varying the expression level, the fusions, if it's C-terminal or N-terminal fusions. And what we try to do is always to have like at least a good dynamic range so that we can measure, deleterious variance and also an increase beneficial, so that at least that we have like a good linear range. But if there's a sub difference on that, that can be fitted by the model. So this can be captured and shouldn't be an issue.
Okay, thank you very much.
Hi, Julia. Great talk and
Thank you.
great work too. I really like the paper. So the what you've shown is what you guys have developed is this PCA, this dual PCA method, which provides a multidimensional readout. And I agree. I think that's really important. I guess my question is, how do you know there aren't more latent phenotypes that are relevant for the function of the protein? And I mean, I think this is just in general a problem. How do we, in general, identify the number of latent phenotypes relevant for a given protein's function? And then kind of how can we confirm that those latent phenotypes are in fact like have these biophysical interpretations that we're kind of here, in suggesting through the specific equation. So I was just wondering, what are your thoughts on this?
Yeah. Well, of course, there can be many things. Like that's why we started. I think the answer would be start simple. Like here we had a very simple model and some simple essays where we know that what can be the main like driving these changes in the molecular phenotype, which is binding another partner. But you can think the same for like enzymatic activity or whatever. The good thing is like stability always tends to be an important one. And we have not only our assay, but there's BAMSEC and many others that can measure that. I think, and there's many ways that we can assay that. We not only need two assays, like someone before mentioned, like we can use like dosage effects, or like this could be another way to having a multi-readout. I would say starting simple, assuming like simple models would be a way, and then once you have a better like intuition of what is happening and see that your model works well, your data is fitted fantastically, maybe add extra layers, have another assay, try to include another state. For instance, here we just use a simple 3-state model, but we could think of a fourth 4-state model where we can have like a bound protein that it's not able to bind anymore, that won't be able to bind. But I think the best maybe is to start simple and then expand from that simplicity when you understand a little bit. something.
Thanks.
Yeah.
Yeah, so Matt Coelho asks, what are your thoughts on predicting how to design small molecule drugs to interfere with protein-protein interactions based on missense mutations that disrupt interactions in your study?
Well, I have to say that I know very little on designing drugs, and the drug design world is not my expertise. But I think this is just, it's a ground map where we can start like thinking on the different, like if we know that mutations affect a certain pockets in the surface, that can be, you know, a starting point, or trying to understand which small molecules could actually bind there and would induce a similar change as these mutations are doing, right? If it's a specific, you know, like charge change or like hydrophobicity change, we can start thinking a little bit more on that. But how to do this in a very systematic manner, like that's out of my expertise, I'm afraid.
That's a tough question. I'm actually going to combine a few questions into one here.
Mm-hmm.
Oh, you want to do? Okay. Yeah, sure. Go ahead.
Beautiful talk, beautiful study. Regarding the allostery, did you have a chance to look at the distance of double mutants? So I'm curious if the second mutation that usually affects the first one, in terms of allostery, does it tend to be in between the core and the dominant allosteric site, or is it usually in the allosteric site?
Yeah, so this is a great question, and I think when we talk about double mutants, and specifically when we talk about the free energy changes due to double mutants, that we cannot say that much, because these are much less confident that these are the actual changes that are happening, because we don't have third order mutants to actually infer very correctly. So I think we need to get the data to get to that question, which would be nice to see some kind of like coupling or interactions between double mutants at the level of energies that could explain, like with this specific epistasis, what's going on. But we cannot say that much
So
about.
you've so far just combined them linearly, assume that most of them will be combined linearly.
The model assumes that, but if anything goes beyond that linearity, that would be a double mutant effect, and we are not that confident that we can claim anything yet with this data. But will come out at some point, I think.
Yeah,
We're now doing combinatorial libraries for that.
Yeah, okay, so this is a combination of a few questions, but were there allosteric sites analyzed with respect to evolutionary conservation? And do you know if any of the sites that you picked up were post-translationally modified? And if not, how would you assay allosteric effects of post-translational modifications?
Wow. The first, if they are conserved, some of them they are, and we know that some of them are enriched for these sites that have been defined as the sectors or like co-evolving residues in this particular domain, which has been well characterized for this. And some of them are enriched on this site, but some of them are not. So maybe these sectors have a bunch of these allosteric sites. If these sites are usually modified with post-transcriptional modifications, I don't know, and that would be a nice additional phenotype to screen in a DMS, definitely.
Thanks. And yeah, the chat is blowing up with questions for you, so maybe...
Okay, I'll go into
yeah,
Slack.
Anyone left over can visit your poster if you're presenting one. Or, yeah, you can go on Vimeo as well and answer that. Yeah.
I just want to add to Justin's comment that there is a method called Lantern by David Ross and Peter Tonner at the NIST, and it can uses the Bayesian method to identify the number of latent phenotypes agnostically. Then you can maybe associate them to known biophysical quantities. It takes you one step further, but not all the way.
Okay, that's good. Thank you. Thank you.
Thank you for a great talk. Thank you for everybody for great talks this morning. And thanks, Moz, for moderating, and Daniel. So now we have time for lunch on our own. And I'll encourage the virtual participants, if you can eat your lunch at the computer and go on GatherTown. I think Jochen said there were maybe 15 people wandering around the virtual poster hall already, hopefully talking to each other. For those of us who are here, I'm going to do a little orientation. So that way is north. And if you go, well, east, just look out the door, and there's a little food court here. That's cheap and cheerful. If you want to go upstairs and across the street and into the lower level of what's called the Hydro Building, sort of a curved building west of here, there's another food court. If you want to do a sit-down lunch, I think there's time. If you get to Baldwin Street before noon, you can usually get a table outdoors. So if you want to try a sit-down lunch, you can go west on College, and then down McCall, and then over on Baldwin. So it's west on College, south on McCall, west on Baldwin. And there's a whole array of restaurants, some with outdoor dining, or just use Google Maps and find a restaurant. So anyway, come on back here at, sorry? So please come back at 1:05, and